Thank you very much for coming. My name is JP, and I'll be talking today about like a big chunk of my PhD project. We're very interested in neotropical fish, and I am very interested in neotropical freshwater stingrays, and I hope that by the end of this talk you'll understand why. And I'll be talking about how we'll be using like phylogeny to predict or to at least get evidence from major biogeographical events in South America. So let's see, the other one, there you go. Whenever we think about like South America and the neotropics, we're thinking about biodiversity, and biodiversity is, I like to tell my students that they are the direct result of the evolutionary processes. So we're looking at organisms that are living in certain kinds of environments, and that over a long time they start to diverge and they're selected over different things. So basically you can think, for example, in two different kinds of water type, bodies of water, have a river, have deep sea, and that environment will act as a filter over some traits or some genetic variation that over time you end up with two different things that are adapted to that environment. So, the other way, sorry. And how does that biodiversity is generated and maintained? So imagine you have a regional population, and then you have a barrier that would break that population in two. I cross geographic because it's not the only one, but it's the most common, is the one that you see like most of the textbooks. And that barrier will create a barrier, isolating the reproduction between those two parts, and those parts being selected for different things. Over a long time, that different genetic diversity that's being selected will cause speciation that relates to the diversity that we see. And we can use that genetic diversity to infer relationships between those parts. You can build phylogenies to see how those things are related, which one is more closely related to each other. But we can also add some information to that those relationship hypotheses, such as distribution, area, geology, even like fossils, to make inferences about their distribution and how old they are and what might have happened that caused that diversification. And we cannot talk about biodiversity and diversification and not think about neotropics. It's one of like those most famous biodiversity hotspots that we have, and especially if you're talking about fishes, it's very important. It's very unique. We have the highest freshwater fish diversity in the world that accounts for 20% of the fish diversity that we have, counting marine and freshwater. So it's like very rich region. And there's a reason for that. When we actually look at the geology of South America, we see that we have a mosaic of different geological chunks, creating this tapestry of geology and environments. We have older areas like huge areas, as you can see here in red, that are cut by sedimentary plains that's carrying sediments from the Andes and Eugene formation here. So we have like different areas. We have a different plasticity. We have a combination of different environments. And why is that very interesting for fish? Because they live in rivers, in this case, or lakes, or lagoons, and that is stuck to the continent. So changes that you have in the continent will affect the rivers and end up affecting the organisms that live in them. There you go. And one of the most beautiful things that we can do is like talk a little bit about biogeography, that we study the distribution of species and environments in geological space throughout geological time. And the study of biogeography with fish is not new, it's not rare, there's a lot of people that do it, but we have a gap in continental level biogeographical studies. You usually have a study that's aimed for a certain region with a certain group of fish. But they talk about that, but it's just like we're taking a snapshot of a certain period of time, and maybe there's some kind of other information that we can talk about. And that's where these guys come from. If you have never seen one before, those are the freshwater stingrays. They belong to the subfamily Phylogeny. Have 4 genera, 38 species. They're beautiful. They're very famous in aquarism, in the aquarium trade, and they're pretty cool because they're found in almost every major river basin in South America. But what makes them especially cool is that they have diverged fairly recently from marine groups. Sister group is the the other subfamily in this family. And we believe that it has invaded this Neotropical freshwater system from the Caribbean around the Miocene, and it's fairly new compared to some other groups of fishes. And what it makes a really good, interesting group to study biogeography is that when we look at these different river basins that we have in South America, we see different number of species and quantity, and even endemics, depending on where you are. So if they are there for this time, we can actually, the other way around, we can ask some questions like how these fish have diversified, evolved the way they did, and what might have caused that diversity in a way that I see differences in distribution patterns and diversity across those different areas. So what we did, we decided to go a little bit ambitious. We did like a big phylogeny with fast evolving genes. We used some priors to date our phylogeny, not only fossils, but we also did some geological calibrations, and then we tried to estimate the ancestral ranges for each one of those clades to see where they're coming from and moving to, and maybe try to understand how they're doing that. So you get this horrendous tree on your left. We can work with something this big, it's not that fun. I tried to make it nicer with colors, but no, it was not easy. It was not, and it didn't work clearly. It's a very bad one. But I'm going to try to point out some nice things that we can see from here. You see that most phylogenetic events that we have, they're younger than 10 million years. They're even younger than 5, which promotes like that, which gives us a hint about a fairly new diversification for this group. We also recover like some interesting paraphyly. For example, we have the genus Lysitrigon inside the genus Potamotragon. And I want you to remember this name, Potamotragon brachyura, because it was going to come up, and it's the sister species for this whole genus. One other thing that we see, when you look in a little bit more detail, we see that most of those clades, they're actually non-monophyletic. You have a lot of nominal species there, forming one lineage. And I'm going to come back to this later in the presentation, but since this is a biogeography talk, I tried to make it look a little bit easier, and I can talk about this for hours. So I'm just going to try to transform this into an area cladogram, and then discuss one of the major things that we can take from this phylogeny about the biogeography of the continent. So the first thing that we do when we do our area reconstruction, and you can see here that the node that gives rise to the freshwater lineages, they're reconstructed to the upper Amazon around the Miocene, which actually matches with the Pebas mega wetland, which was this big swamp that would go from the Caribbean to the Paraná-Paraguay basin, crossing the continent, which kind of like gives a nice support to that hypothesis that they invaded. One thing that we can do in another area of that tree, you can see that we have two independent invasions or interactions between the Amazon basin and the Paraná-Paraguay one. And this is where that Potamotragon brachyura comes in handy, because you see that it's sister to Lysitrigon here, and it's around the end of that Pebas mega wetland. But then when you look at the other Paraná-Paraguay clades, you see that they're fairly younger than that. And that might be related to some river captures that we'd see between the Paraná-Paraguay basin and the rivers in the upper Amazon basin here, here, as well as some of the interactions with lower Amazon rivers in this part of the phylogeny. Well, I'm shifting the gears again, we can actually see that rivers that are now connected around 5 million years ago, around here, they were isolated before, and that is reflected on the phylogeny when you see that taxa that belong to the Negro River here, they're more associated with taxa in the Orinoco, while the Branco is more associated with taxa in the Essequibo than between themselves. And you can see like evidence for like these different faunal corridors that we read in the literature, and we're trying to understand what's going on. Finally, when you look at the lower Amazon clades, that would be the ones that are close to the Atlantic, you see that they all show up in the phylogeny younger than 5 million years, which kind of relates with the new channelization process of the river, with all those distinct river captures. So it's kind of cool, like when you look at a big phylogeny like that, it can be scary, but we have evidence of different biogeographical events that are occurring in different time scales. So you can actually combine those different snapshots of biogeography in one big analysis. But then we can go back and say like, hey, JP, what's going on about all those weird clays that you have with species all over the place? So we try to do a multi-species coalescent analysis to see what was going on, and actually what we recover is that those lineages, they're formed by these nominal species that we have associated with where they come from. So actually like have lineages that are determined by where they come from, not about what they are. And that's pretty cool. But there are some things, some questions that can come, and I know that some people are looking like, about this. Sorry. So before we start like throwing stones at me, we can actually make some questions like, are we using the right markers? Because we know that some genes and some gene trees do not actually reflect the true species tree and the speciation events. We can actually be seeing some cloudy effects from incomplete lineage sorting or hybridization. Or even like, do we have a cryptic diversity that we don't know, have all these wrong names? For example, all these stingrays here, these four are considered as the same four species. For me, that makes sense, but for someone that's not an expert in that group, and I don't like to call myself expert because I'm too scared by that, they're clearly different, right? So something is going on. And I'm going to give a sneak peek on what we're doing now. We're trying to reconstruct a phylogenomics approach to this using a fairly ambitious one too. We have around 10,000 loci, and we're going to analyze species trees and that mixture text and hybridization and structure. But I'm going to give you like a little treat, and what we see here is that even in this way, in our tree, like phylogenomic tree, we see that we have an organization that is related to an area. So you have like Upper Amazon Basin here, the Negro, and another one sister to it with the Tocantins and the Chibu, which are the Lower Amazon. So even from a phylogenomics perspective, we can actually get some insights on distribution, phylogeography, and biogeography, which is neat. With that, I would like to thank everyone that has been helping me throughout the years, and my collaborators, and I know it's not being a smooth ride, and yeah, I could talk about this for a long time, so if you want to talk about later, discuss methodology and other stuff that I have seen, you can hit me on Twitter, you can talk to me at the conference, I'm around, and yeah, thank you very much, and I'll take questions if any. Thank you. Yes.
A lot. So how you can deal with this?
That is actually a very good question. We have discussions with my collaborators. One is here and just like, well, we have all these hypotheses, but how can we actually test on how these kind of like different niches or habitats might be selecting for different forms, or like we have local adaptation. It's something that we're just trying to deal with, because these rivers, as they come from different places, they change, right? And they have different characteristics. So yeah, maybe we can talk a little bit more about that.
I'm working with a river in the south region. I think we can talk a little bit later.
Nice, thank you. All right, guys, thank you very much.